Good morning to the guest of honor, renowned scientists from different educational institutions, audience, and my dear fellow mates. I, Yasha Nirati, presently pursuing Masters of Science from Institute of Bioinformatics and Applied Biotechnology, am delighted to host this inaugural session of Coronavirus
Good morning
webinar
to
series.
the guest.
There have been many such series all over the world, but our main motto is to reach out to those people who do not have a good background of biology or life sciences, such as people in the field of social science, arts, economics, etc. We feel that there is an urgent need to come up with such a program so that people can grasp the basic knowledge behind the prevailing scenario due to coronavirus. There are lots of myths, confusions, and misunderstandings in the whole concept, so we try to pour it into a simplified way from the top experts in this field. I'm honored to welcome our esteemed guest, Dr. Vinod Scaria, Senior Scientist at Institute of Genomics and Integrative Biology, New Delhi. He is a pioneer in precision medicine and clinical genomics in India. He is well known for scientists in sequencing the first human genome in India in 2009. He has established a large research network in the area of clinical genomics with the focus on the rare genetic diseases and pharmacogenomics. His initiatives have helped in uncovering the genetic basis of many genetic diseases in India. Recently, he has analyzed the phylogeny of various strains of coronavirus and has examined the time and place of origin of various clades. I would now hand over to Dr. Vinod, who will be talking to us about understanding the genomes of SARS-CoV-2 in India. Please, sir.
Thank you very much for inviting me here, and a very good morning to all of you. I hope the slides are visible. Um,
Yes,
Over
sir.
the next 30 minutes or so, what I'm going to talk to you is what we have learned so far from the many genomes of SARS-CoV-2 from India, and also draw your attention to the possibilities or rather more open questions which still remain unanswered. Before I talk and start my talk, let me first introduce what our lab works on generally. Um, we are not a virology lab, but we are a lab which calls ourselves doing genome informatics. Now, genome informatics essentially means that we look at genomes of organisms using computers. And you could address a variety of questions trying to look at genomes on computers. One of the major areas we work on is in the area of non-coding RNA or non-protein coding RNA. So these are regulatory RNA which regulates the rest of the genome. We also try to look at how the vertebrate genome is organized and what are the variabilities in the vertebrate genome, trying to understand how diseases could be caused or how the body plan morphology could be changed. We also work extensively with a large number of clinicians across the country on a variety of areas, mostly in the area of rare genetic diseases, where we co-founded the Guardian Consortium almost 6 years ago. A very small part of my lab actually works in the area of pathogen genomes. We used to mostly work in the area of mycobacterium tuberculosis, but of course, very recently with the emergence of SARS-CoV-2 as a major pathogen, it just drew attention to look at the genomes of SARS-CoV-2. Of course, you could find more information about the institute where I come from and our lab at the website out there. Now, to start with, we'll need to first introduce the organism. And here the organism is a virus, a quite small virus called SARS-CoV-2. SARS for the severe acute respiratory syndrome, CoV for coronavirus, N which also stands for the novel. It's also called the CoV-2 because it is distinctly different from the CoV-1 or the first coronavirus epidemic that happened many years ago, at least a decade ago. The viral genome, as we know, is approximately around 30,000 base pairs. which is a quite large viral genome compared to most of the viral genomes that we typically talk about. The commoner ones, which have been extensively studied, for example, HIV, the hepatitis B, are typically in around 5,000 to 10,000 bases, and this is at least 3 times that size. And as we know, we also have read a lot of papers which suggest that they belong to the genus of beta coronavirus and has been found previously in bats in China. Now, all that we know about genomes owes its origin to the first genome that was sequenced and deposited by Chinese researchers quite early in the epidemic. And this was deposited in February, uh, a couple of months after the epidemic broke up in China. In terms of nucleotide similarity, there are around 89% of nucleotides which are similar to the other coronaviruses, including the beta coronavirus subgenes. Now, what I want to draw your attention to is, unlike most large epidemics which happened in the past, there's significant differences and significant learning, at least, that we can take home from this particular epidemic. The first and the most important thing is that, unlike a lot of other epidemics previously, where people were grappling to find the identity of the organism that caused an epidemic, this epidemic was quite distinctly different, that we had not just the identity of the organism, but also the complete genome sequence of the organism. And the first genome, what we call as the WH human 1, or the reference that we use quite often, owes its origin to new approaches to not just identify the virus, but also be able to sequence complete viruses. Now, typically, when you talk about virology and identifying organisms in an epidemic, in olden times, it used to take a quite laborious procedure to isolate the virus, in many ways, culture, propagate the virus, and then be able to identify the organism. But here, you would see this the technology has changed so rapidly that you could change the bodies. You could take the body sample, in this particular case, the bronchoalveolar lavage. You could do a RNA extraction, deplete the ribosomal RNA, and directly go back to sequencing. And this is what we call as shotgun sequencing. So you could take a body sample and you could process the body sample and sequence it directly. And then, of course, use the sequence information to reconstruct the virus and be able to know the identity of the virus. And that's the entire part of bioinformatics. This includes processes like base calling, includes QC, includes assembly, includes QC of the assembly, and of course, being able to put them in the phylogenetic cluster to be able to understand what is similar and what is dissimilar. Now, shotgun sequencing is possible today because of, one, that we have sequencers which can generate large amount of data, millions if not billions of base pairs of data in a single run. And of course, with that comes in the computational power to be able to put together the small fragments, typically 150 base pairs. So small fragments of reads back into assembling the complete organism. And this is what has really changed this particular epidemic, that we could take a sample from an individual without circumventing the entire traditional processes of microbiology, be able to sequence it. And more importantly, after you sequence it, use your bioinformatics capabilities to be able to assemble it back into the complete organism genome. If you think this is quite difficult, it is not. Uh, Very early on, at least in the month of March, we have put together this particular protocol, which students can easily follow. And of course, I believe you could take these links down so that you could go through these protocols. Many of these raw data sets are available in public domain, and you should also be able to assemble a genome with very minimal computational resources. You need an 8 GB RAM Linux system to be able to assemble these genomes. There is also a support group on Telegram, and you could avail this feature. Now, like all pathogens, whether it is viral or whether it is bacterial, the unique property about pathogens is that they evolve, and they evolve by mutations. The only different thing is that the speed of evolution, or the speed at which a particular organism accumulates mutation, is different for different organisms. So, for example, if you know that an individual could transmit a virus to another individual, and and then therefore form a chain, and this chain today we know for coronavirus is approximately around 5 to 10 days. So let's put an average of around 7 days. This particular virus can mutate approximately once in around 10 days. In other words, in every two transmissions, the virus undergoes or accumulates a new mutation. This has a lot of implications, and almost all that I'm going to talk in the next set of slides are going to be uh, central around this very basic concept. And you will see this slide over and over again because this is essentially central to all that we know about virus and virus evolution, not just in coronavirus, but also in all the organisms and pathogens that we see. To give an example, you could see the first Wuhan Q1 genome, which was the first genome to be sequenced and deposited, was collected in December 22nd, 26th. And another genome that was sequenced from a sample collected in Jan 8, you would see that it has accumulated one additional mutation compared to the first genome that was sequenced. And moving forward, in February 27, the virus has accumulated multiple mutations, in this particular case, 4 mutations, which are different from the original strain out there. So this is how the virus mutates. As time progresses, the virus accumulates mutation, and of course, this is also an approach to identify and track back the virus, the origin, and the evolution. Today, we know a lot about the SARS-CoV-2 viruses, thanks to a lot of researchers. Despite being in the harshest conditions, they have ventured out into their labs, collected samples, sequenced the samples, and often they have deposited the samples in public domain so that the rest of the researchers could access it and use it. And this is a very different outcome of the particular epidemic that we are through. Unlike many other epidemics which were limited geographically and was limited to the number of genomes which are available, this particular epidemic, since it is also worldwide, has contributed to a very significantly large amount of genomes which are available today in public domain. To put numbers, we have close to around 60,000 genomes of SARS-CoV-2 in public domain, which is accessible to researchers like you and me. This also includes data which is deposited from individual labs. This includes data which are deposited by large consortia. For example, you could look at the COG, or the COVID Genomics Consortium in the UK, which has deposited, I believe, close to around 26,000 genomes in public domain. And data sharing, therefore, is one of the distinct principles which is different from the previous epidemics of any viral or pathogen origin. Now, of course, when you have a large number of genomes, there's also this problem of being able to access them, being able to search through them, and being able to slice the cake in the particular keywords that you want. And this is one of the resources that we have built quite early on. This is called the Genome Medium, where you could search the viruses which are available by the strain names, by the data source. For example, you could search for sources that come from Delhi. You could search for sources that come from Australia, so on and so forth. You could search by the country name. You could search by the lab which has sequenced it or the lab which has deposited. So there are multiple ways you can search and retrieve this information. I said before that the virus accumulates mutations, and when the virus accumulates mutations, they become a bit distinctly different from each other. And then, therefore, they could form lineages, and lineages are typically displayed on a phylogenetic tree. So, the lineages are otherwise, in scientific terms, also called as clades. So, you could have clades of the same strain. So, for example, the SARS-CoV-2 is one strain, and you could have multiple small clades or lineages, each of them distinctly different from each other by a few genetic variants. And this is also going to be central to what I'm going to talk about in the next set of slides. Of course, you could also build phylogenetic trees and take in all the genomes that are available in public domain, whole or part of it. And there's an open protocol that's currently available, which is put together by one of my students, Bani. It's easily followed, and of course, you could download most of the resources which are also available in open source. Very early on in the epidemic, one of the resources that has come up is this very interesting resource called the Next Train, which came out of the Trevor Bradford's lab. Apart from coronavirus, they have been tracking a lot of epidemics in the past, including, for example, the Zika virus, including the influenza viruses, and so forth. And this is how you could actually look at a phylogenetic tree and look at pathogen genome. So each dot in here is one pathogen genome. This is a limited representation of the 50,000 sequences which are available. And as you see here, they're distinctly classified into different lineages or different clusters, each of them distinctly different from each other by a few mutations. Now, if you take the global data, the lineages could be delineated into 10 distinct lineages or 10 distinct clades. They're named as A1A, A2, so on and so forth. The original isolates from China belong to the B cluster, the B lineage, and the A2A, which is a light blue dot that you see over there, is one of the largest cluster or the largest lineage which rapidly spread across the world. So in many ways, the lineage could also point you to the origin of the virus or the location of the virus from which it came, and could also point to tracing the origin and introduction of the virus in a particular region or a particular community. Now, what is really important to mention here is that practically all the viruses across the world has been transmitted through people traveling on air. So in many ways, looking at a virus genome can also identify which individual from which part of the world actually brought in this infection. And from that point of time, the virus is going to mutate and evolve over the subsequent generations or the subsequent infection cycles. And then, therefore, you should be able to trace not just the origin of the virus in terms of where it come from, or what was the lineage of the virus to start with, and how did it evolve and emerge across subsequent infections in the population. And I'll come back to this again. This is possible because the virus mutates roughly approximately in 1 in around 10 days. And therefore, it allows you to do what we call as molecular tracing or molecular ancestry tracing. Now, this has a lot of implications because it is not just individuals who have come into the country from abroad who have brought in the virus. So that is what we call as an introduction. There are also people who traveled across the country from state to state. This could be people who have traveled not essentially on flights, but also on road. This could be people who have traveled on trains. This could be people who have walked all across the country. But nevertheless, they could all have, at least a small proportion of them could have carried the virus in terms of an infection and introduce them to a new population which was previously uninfected. And looking at the mutation, you can actually trace the origin and travel of the virus. And that's very essential because now you could be able to qualify the transmission across the world in terms of introductions. And now we also able to assess what clade or what lineage of the virus would emerge in a new area, provided we know what the introduction was. Now, coming back to the genomes from India, today we have close to around 1,400 odd SARS-CoV-2 genomes from India, deposited from a large number of laboratories, both medical as well as research institutions across the country. Today covers close to around 21 states and union territories, and of significant mention is the Gujarat Biotechnology Research Center, otherwise known as the GBRC, in Gandhinagar, which has contributed a whopping large number of genomes from Gujarat, close to around 300 odd genomes from Gujarat, which means today we can understand all the epidemiology, all the introductions and the evolution of the virus in one single state at very, very high resolutions. And this is really unmatched in the history of genome sequencing in the country. Even looking at it globally for a very limited population in Gujarat, this is probably one of the largest and the best data sets which span multiple time points across the epidemic of SARS-CoV-2. The obvious question that you would all ask is why sequence over and over again? Why sequence 50,000 times of the same strain of coronavirus? It was sequenced before. Yes, you need to sequence it because one, it allows you to understand the evolution of the virus. You can understand the origin or the introduction of the virus in terms of where did it come from and how is it evolving. And you can also trace community outbreaks if you can understand where did this virus come from and from which contact did it come from. And of course, even more important is that this can allow you to evaluate diagnostics or in some cases even develop better diagnostics and also be able to understand how a potential vaccine or therapy would be efficacious or not. Now, if you look at the Indian coronavirus genomes, uh, we could cluster them into very distinct clusters, as you see here. 6 out of the 10 clusters which are globally prevalent, we have them. And apart from the 6 which are prevalent elsewhere or which have been documented in next strain, we have one additional cluster which you see here in purple. And this cluster is very distinct in the sense that it's a very large cluster only in India. And then therefore we call it as I or the A3I cluster. I, which says that it is a very distinct cluster in India. I'm not saying it's a unique cluster in India, but it's a very distinct cluster in India. And A3I to suggest that it diverged out of the A3 cluster of coronaviruses. Now, the A3 cluster of coronaviruses are present mostly in the Middle East, in the Persian basin. For example, if you say, look at Iran, so many of these green dots that you see here in the A3 cluster are essentially for people who came from Iran. Now, this cluster somehow is an offshoot of the A3I cluster. Doesn't necessarily mean that it evolved from one of these passengers who came from Iran, but nevertheless, it only says that it emerged at some point in time from the A3 cluster. Now, very likely that it didn't emerge in India because there were sequences of the A3 cluster which was found elsewhere in the world previous to the first introduction in India. So therefore, we surmise that this introduction and evolution had happened elsewhere, but nevertheless has become a very large cluster in the country. And this cluster today spreads a large number of populations across the country. And of course, this is not just a picture that I show. You could go back and browse this graph in this URL that's mentioned below. So this is an X-strain implementation of all the SARS-CoV-2 isolates from India. It's quite updated. You would find close to 1,000 odd genomes of high quality on this map today. I welcome you to all to go back and sort of browse this resource. There's a lot of more information that you can visualize on this resource. Now, like I said before, it is the mutations or the variants that distinctly define each of the clusters of the clades. Now, in some cases, the clusters of the clades could be defined by one. For example, if you look at the B cluster, the B clade, it is defined by just one mutation. It could be defined by multiple mutations. For example, if you look at the B1 or the A1A, they're defined by 2 mutations. And in some cases, the mutations could also be shared between clusters. Like, for example, if you look at the I or the A3A cluster, which is defined by 4 mutations, there is a 5th mutation which is also shared quite significantly in frequency with the A3 cluster. So that's what I said, that you could also now look at lineages and sort of map how did this lineage emerge from a previous lineage. And this is exactly one of the reasons why we say that this particular cluster emerged from the A3 cluster, because it has one mutation which is quite prevalent and shared with the A3 cluster. Now, in many ways, these mutations are also mutually exclusive to each other, with exceptions, of course. For example, if you look at the B and the B4 cluster, the B and the B4 cluster shared by one mutation, but the B4 has an additional mutation. And the same is for A3I and the A3 cluster. And the same mutation is also, as you see here, shared by the A1A cluster, and so on and so forth. So in other words, going forward in the future, we might not be able to require a large number of genomes, but now be able to just type these specific mutations which can classify a particular cluster of viruses. So in other words, it might offer a unique opportunity to build cheaper resources which can do large-scale epidemiology and a very, very large number of samples to be able to understand differences between the clades in terms of, for example, looking at pathogenicity, looking at evolution, looking at infectivity, so on and so forth. Now, I said before that the virus mutates, and this mutation rate could be characterized because you have viruses and sequences of viruses spread across time points. So you can quantify over a specific period in time how much new mutations does the virus accumulate. Now, this mutation rate is exemplified by as sites per base pairs per year. And if you take all the isolates of coronavirus in India, the mutation rate is approximately 1.48 into 10 to the power minus 3. So this roughly is 1.48 into 10 to the power minus 3 into 30,000, because you have 30,000 bases in the genome. So that's approximately 44 mutations per year. And that's approximately comes to around 3.7 mutations a month. And that's what I roughly said. It is approximately 7 to 10 mutations per week. Sorry, approximately one mutation every 7 to 10 days. Now, that's for all the viruses, and this pretty much concurs with what you observe globally. But of course, you could take individual clades of the virus and ask this question: do different clades mutate faster or slower than the average? For example, if you look at the A2A clade, which mutates at 1.42 into 10 to the power 3, 10 to the power minus 3, which is very similar to the average of the global mutation rate. You could, for example, look at the I clade, and you would see that this is much lower. This is 1.19 into 10 to the power minus 3. But of course, the A3 and the B4 are very small in number, so we might not really be really accurate in the estimation of the mutation rates. But nevertheless, to state that IA3I clade, which is distinct in India, finds a very small or much smaller mutation rate compared to the average mutation rate of all the clades put together. Now, the next obvious question you would ask is, what are the relative proportions of these clades across the country? And in fact, these clades would have very distinct proportions across samples from different parts of the country. For example, you could look at Delhi. Delhi has close to around 50% of the isolates belonging to the IA3I clade. You could go to, for example, Andhra Pradesh, right here. Andhra Pradesh has close to around 75% of the isolates belong to this particular cluster. You could now, for example, look at Gujarat, which predominantly belongs to the A2A cluster of genotypes. And of course, there are some states, for example, Madhya Pradesh, which also has predominantly the A2A cluster. Now, of course, you would find a lot of gaps in these regions across the country. And what we hope to is collaborate with a lot of laboratories across in these states from which, for example, there are no genomes or there are not much of genomes. This includes states like, for example, Kerala. This includes, for example, Andhra Pradesh, Telangana, the northeastern states, Jammu and Kashmir, Himachal Pradesh, Orissa, so on and so forth, where there are limited representation of genomes, and then therefore we would like to fill in these gaps by adding more genomes out there. Now, from the phylogenetic tree, apparently it seems that this particular clade, which is a IA3I clade, has been introduced once into the country and then spread across the different states, as signified by the relative divergence from a central point in the phylogenetic tree, as you see here. You could also look at temporal proportions of genome sequenced from different parts of the country and ask this question: which clade of the virus is emerging, or which clade of the virus is devolving? And as you see here, the largest number of IA3I cluster came in the month of March and April, and then therefore has been slowly replaced, or is being slowly replaced, by the A2A cluster of the virus. And this is very consistent across the different major states from where we have genomes. This analysis was done by Dr. Sowpati from the CSIR-CCMB, who has extensively collaborated on this analysis. And of course, that opens up a new Pandora's box of questions. And the obvious questions that come to anybody's mind is, of course, if you have difference in the isolate or the genotypes of the isolates, is there a difference in the pathogenicity between the clades? And the pathogenicity is a broad term. This could be much specific in terms of there's a difference in infectivity, is there a difference in disease severity, is there a difference in the rate of immune response or the immune response or the outcomes for that matter. And of course, I stated before that the I clade has a much lower mutation or substitution rate. And the obvious next question is, why is it having a much lower substitution rate? Is it because of very specific phylogeny or the topology of the phylogenetic tree, which makes us miscalculate this substitution rates? Because it is known that when you have a rapid emergence after a single point of introduction, you might artificially have a much smaller rate of substitution. This could also be because these are functional mutations, especially one of these mutation which characterizes this clade belongs to the RDRP gene, which is the RNA-dependent RNA polymerase, which is involved in the replication. So probably this could be a functional mutation which offers the virus a much high fidelity replication rate. And of course, you could access all these resources in terms of the genetic variance for all the isolates that have been sequenced so far in India. Today we have close to around 1500 odd mutations, and each of these mutations have been well annotated thanks to a lot of contributors, volunteers, research institutions which have put the best efforts and contributed to this particular resource. This resource called Indicove is available on the web and it's publicly available. You could Google for it and you could find it. And for each of these variations, we have provided probably one of the most comprehensive set of annotations available anywhere on Earth. So this is contributed by a large number of researchers who put together annotations from multiple different resources from their knowledge base, and this put together essentially becomes a central resource, a central point that everybody can access the variants and their annotations. And of course, for students, there are very many open questions. Open questions are like, can genetic mutations change the efficacy of diagnostics? Can we understand the immune epitopes and how these mutations change the immune epitopes? And then can we evaluate vaccines in clinical trials, or even better, develop new vaccines? And can we understand how these viruses evolve and mutate, and also be able to define functional effects of the functional consequences of these particular mutations? And one of these questions we tried addressing, and it's very pertinent in this particular scenario, because practically all the gold standard tests that we use for diagnosis of SARS-CoV-2 is based on PCR, a very specific type of PCR called the RT-PCR. Now, like all PCR reactions, the efficacy of diagnosis is based on how efficacious is the binding of the primer to the cognate target site. The primer is an oligonucleotide that the manufacturer has designed to identify and or bind to a very specific region in the virus genome and amplify a particular region. And once amplified, it is probed using another set of oligonucleotides, which is fluorescent label, and that's also dependent upon how efficient does a primer or probe bind to the cognate target site. So in other words, theoretically, genetic mutations in any one of these sites which are recognized by the primer or probe could change the efficacy of the diagnostic reagent. Now, of course, this is more simple said than done. That's practically because the primer and probe information for majority of the diagnostic reagents available is not really cataloged elsewhere. Especially even for large organizations, even national organizations, except for example the CDC or the Korea CDC or the Japanese CDC. For many of the national resources, the primer and the probe information is not readily available. So therefore, what we did was to go back and email a large number of companies who produce these reagents and ask the question, what could you share the primer and the probe information? And this was done by a large number of students from Bangalore. Of course, I have the names in the last slide, who put together this very large compendium of around 87 primers and the probes from different companies who willingly shared this information. And we asked a very simple question: for the primers and the probes, do we have mutations in the genome? And if they have mutations, how frequently are these mutations? So the plot that you see here is essentially the coronavirus genome. The y-axis is the variant frequency for variants, the 1500 odd variants in the genome. The ones which are marked red are essentially the ones which fall in either the primer or the probe recognition site. So put these numbers together, there are 123 SNVs and 2 unique indels which fall in at least one of the primer or probe site. There are 13 unique variants which have an allele frequency of more than 1% of the genomes sequenced out there. In other words, these variants could contribute to either false positives or false negatives in more cases for the diagnostic reagents. And as you see here, some of these allele frequencies as high as around 30%. In other words, very unlikely that the reagent that has a particular primer would be working in a country like India. Uh, Apart from this, of course, there are a lot of programs, projects, and data sets which are available in public domain. This is from our initiative on coronavirus genomics. I welcome you to go through this website, and if you have a lot of time to volunteer, please do volunteer. We also have put together a large number of resources to enable researchers, not just in India but also globally, to be looking at coronavirus genomes much more efficiently. This includes resources for phylogenetics. I did show one of the examples. This includes the genomic variant repository, which is Indico, which also I sort of mentioned, and we also have resources which can share genomic information in global standards. This is a genome beacon that we have built with the Dennis Gaur group at Australia. And before I stop my talk, let me acknowledge a lot of people which made this possible. A lot of people who put in effort during the lockdown period at their homes to put together data sets, to put together an analysis. In many ways, many of them contributed wholeheartedly, including volunteers from Bangalore who contributed to putting in, for example, the primer of the probe information. And of course, there are a lot of students from the lab. This includes Bani, Mercy, Kavita, Mukta, Disha, Abhinav, Asta, Mohit, so and so on. Fantastic collaborators, Sridhar for most of the NGS that we do, and fantastic collaboration from CSIR-CCMB, which is a sister lab, which also has extensively invested in looking at coronavirus genomes. And we are deeply indebted to a lot of researchers from across the country who have put together the genomes from the respective states and made it available in the public resources, either the GenBank or the GSAID datasets. And of course, this is all possible with funding, and funding in almost all instances came from CSIR. I think I'll stop there and take questions. But before that, let me also use this opportunity to invite you to participate in this social endeavor to understand COVID-19 disease. So this is a web app which allows you to track the progress of COVID infection. So this is essentially for people who are not yet infected. You could enroll yourself to this particular program, and on a regular basis, we would get back to you asking very specific questions, trying to understand what are the risk factors, what are the comorbidities, and also assess what are the outcomes of infection. And this is under the assumption that a significant large number of people, in our assumption, close to around 20 to 30% of individuals in India would get infected with coronavirus at some point of time. And this data and a crowdsourced approach would allow us to learn a lot more in not just understanding the pandemic, but also in many instances to be able to be prepared to the next pandemic. I think I'll stop there and take questions. Thank you.